So, welcome everybody. So, as you can see, Philip, you've got a bit of a live audience. Some of whom were expecting you in person, but it's a pleasure to welcome Philip. That probably doesn't need much introduction at all. Philip's statutory professor of mathematical biology at Oxford, fellow of the Royal Society, and numerous other accolades. And so it's a pleasure to welcome him. And I've forgotten the title, Philip. Was it collective cell motion or modeling?
Yeah, collective cell migration. Yeah,
cell migration. Okay. So floor's all yours.
Okay. Well, thank you, Siv, and thank you for the invitation to this. And I'm really sorry I can't be there in person. I'm just sort of overwhelmed at the moment with a lot of sort of bureaucratic stuff. So I'm really sorry that I can't be there, because it would have been great to see you all in person. And I'm going to talk about two examples of let's get this up. Well, first of all, about collective movement in general. So we've all seen collective movement, like whales, fish schools, flocks of starlings, and this sort of movement happens right from the organism level right down to the level of cells. And I'm going to focus on cell movement. And in that context, one can think, roughly speaking, of two types of cells. There are epithelial cells. These are cells in sheets, and you can have the collective movement of sheets, for example, in epiboly, where in early development, sort of epithelial sheets move over the embryo. Then you can also have cells within a sheet moving. And then there's also the loosely bound cell movement in mesenchymal cells, and that's what I'm going to focus on. And I'm going to talk about two examples: cranial neural crest cell migration and cancer cell invasion. So the neural crest stuff, that's work with Paul, Glessa, and Rebecca. And in fact, Paul's just moved from next term. He'll move from the Stowers to Notre Dame, and with a number of colleagues in the department and a series of graduate students. And so the neural crest is basically what happens early in development is that cells sort of delaminate from the neural tube and undergo what's called epithelial to mesenchymal transition, EMT, which is also what cancer cells do. And then these neural crest cells migrate and then form structures. And there are four neural crests: cranial, trunk, vagal and sacral, and cardiac. And here's just a cartoon. Here's the head, tail, backbone, and then here's these cells moving off in chains to different areas. And so here we have them sort of moving around like this. And it's important to study this because, in fact, nearly 1/3 of all birth defects occur as a result of something going wrong with the patterning of neural crest and the vasculature. Another reason for studying it is that neural crest cells are very similar in behavior to melanoma and neuroblastoma, highly aggressive cancers. And it serves as a very powerful and useful paradigm for invasive movement in general, and is experimentally tractable. And in this review article, we've sort of summarized here what we see as the mathematical challenges in dealing with this problem. And I should mention that lots and lots of people work in this area, and this doesn't even begin to list the number of modelers who work in this area. And so before I talk about the cranial neural crest, just to put it in context, here we have work on the gut. And this is by Kerry Landman and Matt Simpson collaborating with the biologist Don Newgreen. And the thing that happens in Hirschsprung's disease, this is a disease in which so in the gut, the neural crest cells, they have to reach the end of the gut, and the gut is growing. And if they don't reach the end of the gut, this leads to problems with the gut that ends up resulting in large deformations of the gut. And it's natural to think that if these cells aren't reaching the end of the domain, there must be something wrong in their movement. And what they did was they developed a mathematical model, which is essentially building on the classical Fisher equation for cell movement. And we know in the Fisher equation, movement depends on diffusion, but also depends on proliferation. And what they did here was by looking at what would happen if you did implants between quail and chick. So then you would have two different cell types, N1 and N2. Here's the diffusion. Here essentially is the proliferation sort of generalized now because you've got N1 plus N2 instead of just N1. Here, this term arises because you're modeling a growing domain where V is velocity. And what they found out was that to get full colonization, it depended not only on the diffusion, but on the cell number. And the cell number in turn depended on the proliferation rate. So this gave a new insight that it could be that the cells are moving perfectly fine, but the problem is in cell division. And then here's another example. This is in from Roberto Mayor's group. This is in Xenopus, the frog. And here they have this idea of co-attraction and co-repulsion, which is called here CIL for inhibition of locomotion. And the idea here is that you've got the cells, one cell attracts another cell, but then when they get close together, they inhibit each other, they repel. And so you get this movement. And they model this with this quite complicated individual-based model where they actually put in mechanics and various things like this. And what they showed here was if you had both inhibition and co-attraction, you got the movement. If you missed one of these, you didn't get the movement. And what you're looking at here actually is a pulse, essentially, of cells moving. And in a more general context here, Kevin Painter and colleagues looked at this with having a non-local interaction term here. And this can be considered a generalized case of having something like a Leonard-Jones potential, where you have short range repulsion, long range attraction. And what they found is that, so this is time, this is space, and then you could get these patterns to form in one dimension, as well as getting the sort of movement that's observed in the Xenopus. So moving back now to the one we're looking at, we're in the chick, and we're looking at the movement of cranial neural crest. And here's the head, here's the tail. And these cells go, and then when they get to, this is called branchial arch 2, that's where they differentiate and form the structures of the lower jaw and upper neck. Now, these cells, as they move along, don't divide. So it's not a proliferation-driven movement like in the gut. Moreover, it's known that they respond to signaling cues. So it's not a self-contained movement like in the Mayor model for Xenopus. And so here we have data, the domain is growing, Here is the length of the domain essentially against time, and over a day, it grows from, roughly speaking, 300 microns to, roughly speaking, 1200 microns. And it sort of looks like a logistic growth. So what we're going to do is we're just going to fit a curve to that growth. There's a bit of curvature. The curvature is quite small compared to cell size, so we're going to ignore the curvature. The depth is about 3 cells thick, so we're going to ignore that. So we're going to consider a fat rectangle that's growing uniformly in space in the x direction, growing like this in time. Cells are going to come out of the neural tube, and basically they've got to get to the end of the neural tube in this coordinated chain type movement. And the model hypothesis, the prevailing model hypothesis, was that there was a chemoattractant, VEGF, produced by the overlying ectoderm, and could that be sufficient for robust invasion? So what we did was write down a reaction diffusion PDE for VEGF, and I'll mention that a bit later. We need boundary conditions. Now, there's known already 6 years ago that in Xenopus, this thing here was present, versicin, which acts as a repulsive thing so that you could have zero flux boundary conditions. When we started working on this, there were no chemical signatures that our experimental colleagues were aware of. So we worked with different types of boundary conditions, and we found that the key results that we're going to mention, whether we had Dirichlet boundary conditions for the VEGF or zero flux boundary conditions, they didn't change the results very much. But I'll come back to talk about that later on. And we consider the cells as being discrete entities, and they randomly send out Philopodia, sense the chemical concentration in the surrounding area. And if the chemical concentration is larger than at the center of the cell, accounting for noise, they will move up the gradient with a constant speed. And here's a model for the chemoattractant. So because it's growing in the x direction and not in the y direction, we rescale 1. So this is 1 over L squared, where L is given by this, just fitting to that logistic curve. Here's a production term of the chemical. We just put in logistic growth here. The results are quite robust to the form of that. Here's that dilution term, because we're in a growing domain. L dot is d by dt. And here we assume that the cells at xi, yi consume the chemical with a Gaussian consumption rate. Doesn't really matter what the form of this is. And then what we find is that that verbal model doesn't work. So here we have the domain, and as the domain grows, what we find is that because the gradient created by the cells is local, we actually get a pulse of cells moving, and these cells get left behind. So we don't get the coherent movement. And in fact, if we go back and think of something like the Keller-Siegel model, which you could argue is a sort of coarse grain approximation of this type of model, where n is cell number, c is the chemical, what you find is that if you have boundary conditions of n is 1 at one end and is 0 at the other end, then the only way you can get the sort of classical Fisher type traveling wave is if you have a singularity in the sensitivity, which sort of means that as the chemical concentration decreases, you become infinitely sensitive to the chemical concentration. So it's known that this model can only exhibit a sort of pulse type movement, the sort of thing that we see in Xenopus, but not the classical traveling wave. But in fact, in our example, we don't have the boundary conditions of the classical Keller-Siegel model. And so we have a student at the minute working on this to see, are there conditions under which this could exhibit a traveling
Can I ask you a
wave?
question, Phil?
Yep. Mhm.
So in Keller-Siegel, there's this quadratic nonlinearity, and in your equation, N1 was multiplied also by 1 minus N1 and 2, also quadratic nonlinearity. In Keller-Siegel, in chemotaxis, the main issue is blow up, finite time blow up, right? Because of quadratic.
Yep.
Now, how about your model? How do you rule out blow up N1? I saw this term N1, 1 minus N1 plus N2. There's also quadratic term in your diffusion equation. Does blow up mathematically exist? Is it biologically relevant, like high concentration of something like in chemotaxis? That is my question.
Yeah, so here, because we have this term in the chemoattractant, we don't get blow up.
No, this is, I understand, this logistic terms is like a KPP,
Yeah.
but I saw early equation with N1 and N2, it was N1 times minus N1 plus N2, something like that. Yes, and that's, okay, yeah, here, so this
Oh,
is,
yes,
this is
this
a quadratic term.
Yeah,
The second
but-
term, this term is quadratic, so it may be like in Keller-Siegel lead to some blow up or something.
yeah, but this isn't a Keller-Siegel model. This is a Lotka-Volterra model, so there's no.
No, I understand.
chemoattractant
It just it has quadratic term. Never mind then. Yeah.
Yeah,
Like x dot
so-
equal x square can lead to blow up, right? It's not a Keller-Siegel model, but once you
Yeah.
have quadratic term, then there is always a danger of blow up, and you have to do something to rule it out. It does
Yeah, well,
not have
this-
to be cross diffusion term, but it could be. Yeah.
Yeah,
That is
so
the
I don't know,
question.
because I'm just mentioning this as work that other people have done. I don't know if
Okay.
they've explored this. I
All
haven't worked on this model, so I
All
don't know
right.
if they've
Okay.
explored
Okay.
this.
Okay.
Yeah.
Okay. Never mind then.
Yeah,
Yeah. Thank
okay.
you.
Yeah, sorry about that.
No. No.
Um,
No. That's good.
Okay, yeah, so if we move along with here, then we find that this doesn't work. And so we're still pursuing this sort of thing here to see if we can understand what's going on. But then the first student who worked on this, Louise Dyson, she proposed that if these cells at the back here, if they've got no chemoattractant to sense, what happens if they just sense the cells at the front and stuck to them? And then they would be dragged along. So she proposed, could there be two cell types? One that senses the chemotactic gradient, moves up the gradient, the other that sticks to the cells at the front. So then we asked our colleagues, I mean, this is just summarizing what we said, and don't need to look at all the detail of this. They basically found significant differences in gene expression of the cells at the front, at the back. In particular, they found that certain cadherins that are the hallmark of stickiness were upregulated at the back. And of course, it's more complicated than that. Biology always is. If you go from back to the front, you find that there's genetic changes happening everywhere. It's not just a case of two cell types. So one of the things that we're exploring at the minute, and this is work with Jose Carrillo, is the idea that it looks like there could be a gradient of stickiness along here. So we're in the process of exploring that at the moment, but we don't have any results on that. So of course, that stickiness is one possibility. Another possibility is maybe the cells at the front are secreting a chemical. Another possibility is that maybe the cells at the front create a tunnel. And that makes it easy, because remember, these cells are moving through a matrix. Maybe the cells at the front bore a tunnel through the cell, and then the cells at the back, if they get into the tunnel, it's easier for them to move. So we'll come back to that. We did quite a lot of experimental validation with the previous model. I'm not going to go into the detail of that. We sort of did things like what happens if you remove cells or implant cells, et cetera, and then compared those with the model. I'm not going to go into that because I don't have time. Instead, I'm going to just mention a couple of things. One question we were asked was how many leaders are necessary? Now, because our model is not calibrated with experimental data, we can't make such quantitative predictions. But what we can do is make a qualitative prediction of what happens if you change the number of cells, if you increase them or decrease them qualitatively. So here's an example here where we count the number of cells as they invade. Plot the diagram here. This is distance along the corridor. And these are the normal ones. And these are the ones where we increase the number of leaders. So the model predicts that if you increase the number of leaders, you will get invasion, but fewer cells will invade. So they did the experiment with something called HAN2 that upregulates the genes expressed by the leaders. And what they found, here's the normal, and here's the HAN2. So you get invasion, but fewer cells. So qualitatively, exactly the same as the model predicted, quantitatively, not the same. Look here up to 8, here up to 16, but we didn't expect this to be quantitatively the same. Another thing to point out is that I glossed over this problem here, that what happens if the leaders get too far away? Then the followers can't catch up with them to stick to them or respond to their signal. So this suggested, is there something that controls the speed of the leaders? So what Paul and his colleagues did, they looked, and here's the neural tube. They found expressed right next to the neural tube is something called DAN. And DAN is a protein that reduces cell speed. And so what happens, they also noticed that as the neural crest cells move through here, the DAN is no longer expressed. So what that means is the cells at the leaders face this barrier that they've got to go through, and the followers don't. So this could be slowing down the leaders, but not slowing down the followers. As I mentioned, neural crest and melanoma have lots of things in common. So the obvious question now is, sorry, here's just the experiment where here's a neural tube, cells coming out of the neural tube, and what Paul and his colleagues did was put strips of DAN. And what you see here, there are more cells on the strips without DAN than there are on the strips with DAN. Do the same to melanoma, and you find that melanoma don't like the DAN. So the DAN could slow down the melanoma. Okay. So now we go on to the question about tunneling. Is it possible that there's tunneling going on? So that led our experimentalists to look at the leaders, and what we found was that the leaders upregulate something called aquaporin, which is a water channel. And what that does is it sort of creates an inertia, so to speak, in the cells, which means that instead of the cells randomly sensing the gradient, the filopodia, the sort of fingers that they protrude, are stiffer and more stable, which means that when they sense some gradient, they move longer in that direction before they sense again. Moreover, this creates tunnels. So what they did, and I'll try and see if I can get this to work. I think I have to come out of sharing and then go back in again. Oops. Yeah, sorry about this. So wait for that to go away. Yep, slideshow. Oh, from current slide, sorry. Oops, no, I haven't managed to get this away. So I was trying to get rid of this. Right, that's it. Okay, so what they did now, so here's the neural tube, here's the cells, here's normal, here's where they inhibit the aquaporin, here's where they enhance aquaporin, here's the model where we do the same thing. And so the black cells, they're the leaders, the yellow cells are the followers. And what you can see here is the inhibitor, as you would expect, don't go as far as these ones. Here you see when you overexpress, you get really long bits of movement and a breakup of the whole thing. And that's what's observed here in the simulation. So now I'm, sorry, is there a question? No. Okay. Now moving on to the boundary condition, which I mentioned that we didn't really know what the boundary condition should be. So what we decided to do, and this is work that's presently in revision, we said, well, look, here's the corridor. And belowhand, before we had some boundary conditions right along this corridor. But this is part of a tissue. The tissue spans in the y direction beyond the corridor. So what we said was, well, suppose you had a boundary, an internal boundary condition, maybe just to here, then what would happen? So what we did was we said, well, if we'll partition the domain into 3 regions. If all the cells are in this region, then that's good invasion. If the cells go into this region, then it's partially confined. If the cells go into this region, then it's really bad invasion. And so what we did then was we said, right, let's impose zero flux boundary conditions all along, and you get really nice invasion. Let's have no zero flux boundary conditions along here, but have them along here. The cells go all over the place. Let's have partial confinement, and you get what you would expect, partial confinement. And we did this with the sticky model and also with tunneling model. And what we found, if you look here, these are stochastic simulations, so we have to do lots of them. This here is the mean fraction of cells that are not in a chain. So if this is high, it means it's really bad invasion. And what you can see, the mean, and now along here is the length of the internal confinement boundary. So what you can see here, there is a lot of noise going on here, but if you look at the mean, at roughly speaking 500 microns, the mean collapses to 0. And this is for one of the models. This is for the other one. Roughly speaking, 400 or 500 microns, it collapses again. So what this is telling us is that as we only need partial confinement for invasion to occur. So this motivated our colleagues to look right next to the neural tube and see if they could find a chemical that inhibits neural crest movement. And what they found were these two things here, one thing called collect-12 and one thing called trail, and they inhibit the movement of the cells. And they are expressed in the first part of the domain, but not in the second part of the boundary. So these, we think, are the ones that are giving the partially confined domain, as predicted by the model. Now, in all of this, we've ignored mechanics. And what our biological colleagues told us was that as the cells are moving along, there's something called puncta, fibronectin puncta. So these are like little parcels of fibronectin. And fibronectin is something that gives signaling cues to cells. And what they noticed is that as the cells move over these parcels, these lumps become aligned as fibers. And so the question was, could that be providing the signaling cue for the cells at the back? And so what we've done here in a simplified version of the model, and I'm just showing you one simulation, these little spots are the puncta, then here's the leaders, and as the leaders move over the puncta, they align them, and then as the followers come along, they've got more chance to move along the aligned fibers. And so I haven't put any of the mathematical equations along here, but basically what you do is you have a non-local sensing area. You basically take the mean of the cue in that local sensing area, a bit like the classical Vichek model, and you move in that direction. So that's what you do. And what you can see here, this is with no internal boundary. This is really nice invasion. And it's stochastic, so sometimes you get really horrible invasion. In fact, the funny thing is, sometimes experimentally, you do see this happen. And this is one thing we're exploring at the minute, is how does that happen?
So mathematically, it would be free boundary domain. It would be a domain with free boundary, moving boundary.
Well, here it would be that if you got to here, it would be zero flux. So if they got to here, then we would just have them bounce back off. But you can see that essentially there's no boundary effect as they move along here. So we haven't actually carried this out till when they reach the boundary, but we would have them just bounce back off the boundary.
Okay, all right, thank you.
Yeah. Okay. So what Duncan did, because of the parameters in this model, he assessed some of the key aspects that other people have looked at, namely contact guidance, which is where as the cells move, they align the matrix, and that allows the followers to move. Haptotaxis, this is movement up stickiness gradients, and it's known that fibronectin is sticky, so that's something to bear in mind. And then volume exclusion. Remember, these are not point particles, these are cells. They can affect each other, they can sort of push each other out of the way. And what he found was that volume exclusion can hinder migration, but it can also enhance migration. It depends what else is going on. And by doing a very detailed statistical analysis, lots and lots of stochastic simulations and everything, what he found, this general result, which is that contact guidance and matrix remodeling, I should have mentioned that, I mean, that's basically as the cells move over the haptotact, the puncta, they remodel it and align it. What he's shown is that contact guidance and matrix remodeling are sufficient to enable collective migration. And this is something that our experimental colleagues are going to test in the near future, hopefully. But what this doesn't do, as we saw here in the background here, is that this movement happens. But if on top of this, we were to put the chemotactic gradient, which gave the cue to the leaders to move in the horizontal direction, and maybe also had a partially confined boundary, then that together with this here, we think would lead to the sort of movement we see experimentally. So there's an example of sort of leader-follower cooperative behavior. And just briefly, as we're running out of time, just talk about cancer cell invasion. And this is work with a former student, Maxi Strobel, and colleagues at the Moffitt Cancer Center, Bob Gillies, who very sadly passed away a few months ago, and Sandy Anderson. And what we did here was, instead of the sort of notion that people have about cancers, you've got a cell that's a generalist. It can do everything it needs to do to invade. What we said was, let's suppose that we've got two barriers to invasion. So one barrier is the extracellular matrix. So that provides a physical barrier, and it's known that cells have to degrade the matrix in order to go through the matrix. And let's assume that the normal cells are competing with the cells for nutrient, and therefore stopping them from growing. So there's two barriers there. Matrix, a barrier to movement. Stroma, a barrier to proliferation. So there again, if you think of the classic Fisher KPP model, you need movement and you need proliferation. This is stopping the movement. This is stopping the proliferation. So now we said, suppose we have one cell type, we call it TA, tumor type A, that produces acid, lactic acid, which is toxic to the stroma. So it kills the stroma. So that frees up the problem with proliferation. And let's suppose then there's another tumor type, tumor M, tumor matrix, that degrades the matrix. So that frees up the diffusion part of the thing. Then what happens? So here's an equation, pretty obvious sort of equation. Stroma cells, logistic growth, I mean, this has all been normalized. This is lactic acid. It's produced by the acid cells, and it kills the stroma. The acid cells, these are all competition, Ludwig-Volterra competition. And then this is movement up. This is diffusion, but where the matrix is hindering the diffusion, but the TM cells degrade the matrix and reduce that. And then we get traveling waves. Not going into the detail. We can do the standard traveling wave analysis. And in this particular paper, there's a lot of degeneracy going on here. So we kept away from that degeneracy in this paper. And what we found here, and this is sort of illustrating all our traveling wave analysis in the cartoon, that if you have mutual coexistence, these results aren't that surprising. If you have both cell types, the Lotka-Volterra give you mutual coexistence, you get invasion. If you have dominance of the matrix degrading cells, you get just a stop after so often because there's no room for proliferation. On the other hand, if you have dominance of the acid-producing cells, you do get a temporary cell invasion before it stops. And then you can also get this behavior where you get spatially mutual exclusion of the two tumor cell types. And this is not a proof that this model is correct. It's a sort of something that says, well, maybe this is worth looking at. This is in human breast cancer, done by the experimentalists on the paper. And the green cells are the cells that produce the acid, tumor cells that produce the acid. The purple cells are the cells that degrade the matrix. And what you can see is they look like they're sort of mixed up, but obviously we would need to do a lot more experiments and a lot more detailed investigation of this to see whether this has anything to do with what's going on here. Now, I mentioned the degradation part and how you then get degenerate diffusion, cross diffusion. So what we did was take a very simple version of this, and this is work with graduate student Chloe Colson, former colleague Faustino Sanchez-Gordonio, Helen Byrne, and Tommaso Lorenzi. Here we have cells degrade the matrix, that creates room for division, and then we have normal tumor growth. And what we find is that there's a lot of degeneracy in this system. Moreover, if you look at here, there's a continuum of steady states. And I won't go into all the detail of what we did here, but we converted to traveling wave coordinates in the classical way, and then convert this to the three equations as usual. And what we find is that we've got the classical sort of almost like the Fisher type, Fisher KPP type, or Keller-Siegel type boundary conditions. But then we also have this continuum of steady states far ahead of the wave. And we do various shooting arguments. Here, the continua of steady states. Along here, we're trying to go from here up to here. And you can see here, the ODE sometimes gives solutions that are not viable because they've got negative values here. And so by doing shooting arguments, what we ended up proving was the existence of traveling waves from n equals 1, m equals 0. So that means all cells, no matrix, were as far ahead of the wave, no cells, all matrix. And these seem to be unique for any positive wave speed. However, if we allow far ahead of the wave for there to be some matrix, then we find unique traveling wave for any wave speed, and we conjecture that it's greater than or equal to a strictly positive minimum, and that positive minimum is really just an extension of the classic Fisher KPP model. But we don't have a proof that that's true, but extensive numerical simulations suggest it's true. but we haven't nailed down the proof of that. And so what we see here, again, something that's not particularly surprising, is that by having these simple non-linearities, the problem becomes, it actually became a lot more complicated than we thought it would. If we were naive when we started looking at this, we thought this would only take a few months. It ended up taking like 2 years to deal with this problem. So I'll end there and just show you there that what we're doing in these two different models is the idea of different phenotypes and how they interact with each other, because there's heterogeneity in cells and different cells can do different things. One thing I haven't mentioned in here is that cells can switch phenotype. And that's what we're looking at the minute, is can these behaviors that I mentioned here be made much more robust if you allow for phenotypic switching? And that's something we're looking at the moment. So I'll stop there and thank you for your attention, and happy to take any questions you may have.
Well, thank you, Philip. I know we've talked about different aspects at various times, but
yeah, that's right,
when every time you do the leader and the follower cells, and today when you mentioned the cadherin expression, I remember a long while ago seeing the papers on E-cadherin as an indicator of mesenchymal to epithelial transition. And I'm sure the real scenario is a lot more complicated, but do you think the leader follower thing fits into this epithelial mesenchymal type framework? Because when you get metastases and things like that, it's the epithelial to mesenchymal that sort of leads the metastases. It's sort of and then when you get to a distant site through the blood vessels, it's the mesenchymal epithelial transition that gives you the secondary metastases. And I was just thinking, it seems like a strong analogy between say neural crest cells and the leader followers, and and epithelial cells do express a lot of the cadherin, and are sort of fairly stationary, right? So I was wondering if the analogy would carry forward to this sort of epithelial mesenchymal framework as well, for say thinking, yeah.
I mean, I think they're very, very strong analogies. So when these cells delaminate from the neural tube, so they're epithelial-like, they're on the neural tube, and then they come off the neural tube. So they have to go through an epithelial mesenchymal transition to come off the neural tube. And then when they get into the branchial arch, then what they do is they differentiate and they start to divide. They start proliferating so that they can form the structures that are in there. But also, I mean, what we've found, or what experimental colleagues have found, is that different cadherins are expressed along different parts of the neural crest. So the leaders also upregulate a cadherin, but it's less sticky than the cadherin that's upregulated by the followers. So that's why we're now looking at having a gradient of stickiness to see what would happen. But another thing going along in the analogy is that the leaders, again, which was found by our colleagues, upregulate genes that produce enzymes that degrade the matrix. And these are precisely the same enzymes that the tumor cells, which also have a leader-follower behavior, that they upregulate to degrade the matrix. And also, of course, these neural crest cells are responding to VEGF, which is precisely the chemical that tumors secrete into the environment so that endothelial cells will go to the tumor and form blood vessels and carry nutrient to the tumor. So I think there's a lot of analogies
Right.
between the two. And what I think the powerful thing about the neural crest is that it's tractable experimentally in a way that the cancer, a lot of the cancer stuff isn't yet, because the cancer stuff is so much more complicated.
Yes. Yeah, in fact, I noticed Kathleen's taking the microphone, but when you mentioned the aquaporins and things, I remember some of your work on hydrocephalus and things where you looked at.
Yeah, that was a long time ago.
Yeah.
So I'm going to have to switch the light on here because it's about to go off. Sorry, go ahead. Yep.
So my question goes back to your first project where right at the start, you sort of determined that your leader cells were going to just absorb all of this VEGF and you'd have nothing traveling past them. So I was wondering, did you look at what if those cells only uptake VEGF in a cyclical sort of time, like maybe they take in a bunch and then it takes them 24 hours to process it so that you could have sort of waves passing, behind them and pulling, so it was more of a dynamic pulsing of the VEGF gradient that could pull the cells along the tunnel or your path.
That's a good point. We haven't looked at that. One thing is that when experimentally, they have some images which seems to show that VEGF does disappear near the neural tube. And also on the time scale of this, I mean, it takes them about a day or a day and a half to do the invasion. So we haven't looked at that idea, but yeah, that would be a very nice idea. Yeah, if they just took up some of the VEGF and moved, then yeah, I never thought about that. That's a really good suggestion to look at, because then again you might have that in the end the VEGF does get degraded, but by the time it's got degraded at the back, a good few cells have already got in and are moving along. Yeah,
Cool.
And I mean, another thing that fits in, which I didn't mention, is that in all of this, we've assumed that the domain growth is uniform in space, but it's actually not. We found out recently in a paper that we published with Hans Othmer last year or something, that there's different growth dynamics going on. And so I think if you coupled that with this sort of cyclic use of the VEGF, then that you might actually get that to work. Yeah, I'm not going to be able to sleep tonight now because I'm going to keep thinking about that. That's a really good suggestion.
Oh, I'm glad you think so.
I mean, the non-local effects, I was just thinking, are sort of quite pervasive in all of these things, right? When you mentioned, because I was thinking even in the context of radiotherapy and stuff like that, I remember these sort of bystander effects where you sort of, you're going to radiate one, but then a cell somewhere else actually responds to it. So maybe, are there any other questions?
So I've never studied this type of model before, but I'm a little bit familiar with models of synchronization in populations, like the Kuramoto model.
Yes, yeah.
And one thing that can happen, getting a bit of echo. One thing that you can have happen there is that at small population sizes, you get very exotic behaviors, and then everything gets kind of, generic and synchronized at larger population sizes, like you pass to the continuum limit. So I was curious, just in the types of models that you study here, is that something that you ever see? And also in experiments, like kind of, if that line does exist, what side are you really on in an experiment? Is it more finite size, or are you closer to a continuum kind of behavior?
Yeah, these ones, I mean, we did start off looking at continuum models of this, but they were really sort of sort of variations of Fisher Keller, not Fisher KPP, of Keller-Segal, and we couldn't get the sort of traveling wave that we wanted, and then we didn't pursue that very much more. And the sort of leaders, I mean, like in that diagram where I showed where they did the experiment of what would happen if you change the number of leaders, you've got of the order of 8 or 9 cells as being leaders. So it's a very small number of cells. And therefore you are quite away from the continuum limit. Whereas with the followers, there is a possibility of having a continuum limit. And in fact, what Duncan Martinson, who was the graduate student, who's now a postdoc with Jose Carrillo, is doing, is now coming up with a hybrid model where you have discrete linking, discrete leaders linking to a continuum of followers, and looking at that to see whether he can capture the behavior of the agent-based model with that sort of approach.
Gotcha. I see. Okay, that's interesting. Thank you.
Yes, I mean, generally that sort of hybrid type model is something that in this context and in any sort of modeling of cancer, etc., is something that long term you'd like to get, right? How, if you do the sort of discrete agent-based modeling, how do you, the question of how information gets transferred up to the continuum level.
Yeah,
right?
that's right.
Yeah. But maybe I can just ask a general sort of philosophical question, just if anybody else doesn't have, I mean, the analogy that early on in math and biology that was made, comparing it with math and physics, and how physics sort of really took off when he came in with these sort of mathematical approaches and the fundamental principles that drive physical systems. Do you see any possibility that, say, with math and biology, that that we still have to, there are maybe some fundamental principles that drive development and growth, which are not just simply the physical laws, and how long do you think before we have the Einstein of mathematical biology who'll come up with a completely different, I sort of wonder sometimes whether we need a completely different perspective, or whether this sort of PDE continuum type approach. I mean, I know Ryden Twarek, for example, has come up with completely different ways of looking at viral capsids, and what are your thoughts on that?
So I totally agree with you. Many years ago, Dennis Noble, who I'm sure you know, he used to have like a lunch sort of journal club thing, and I was one time giving a talk about reaction diffusion theory, and then how there's certain models like Drosophila that disagree, that form patterns in a much more sort of not as elegantly as the reaction diffusion. The Turing model predicts you can get these nice patterns, but in Drosophila, what you find is that there's a system for each stripe that you observe, and it's a hugely complicated system, and that how that nature doesn't make models in an elegant, doesn't make patterns in an elegant way. And Dennis's response was, but if you had the right mathematics, maybe it would be elegant. And I think that's what you're saying, that if we could somehow think about this in the right way, it would all make sense. And I strongly believe that, because if it really was so complicated, I fail to understand how any of us even exist, because something would have gone wrong.
Yeah, I mean, you're absolutely right, because I remember early on when we were working with Jim Drake and people, and we had these very, it's a bit like Occam's razor. You have a very complicated problem, and And, you know, we have this very simplistic model. And I was always a bit shocked how, you know, when you ran it computationally, we actually, you know, Jim would give us these CT scans before and after insertion of a shunt. And the simplistic model, you know, you get, you know, pretty much, you know, close to what you see in practice. And so at that stage, I started thinking, you know, sort of similar that, you know, as a physical organism, why have we persisted so long? You know, it must be because there's some robustness in, you know, you can sort of throw a lot of things out and still, you know, you get close to where things are. Yeah. So
Yeah,
it's,
that's right. And funnily enough, that robustness
Yeah.
that helps us to survive is
Yeah.
also then the robustness that comes back to haunt us, because then in something like cancer, where you target one aspect to try and kill the cells, and it just uses a different way around to get
To
around
avoid,
the problem.
Yeah. No, that's very interesting. Any other questions? Okay. Well, Philip, I guess is it no, it's not quite dinner time for you, but I guess
No,
you
but
probably have to
getting there,
think about dinner. Anyway, thanks a lot. And
well, thank you,
hopefully-
and thank you for the invitation. And it's a fantastic discussion, this, and I really do wish I was there, but we could talk about it.
Yeah, well, we'll hopefully get you out now that things are opening up everywhere, you know, so
Hopefully, yeah.
yeah, okay.